So the goal for today is to wrap up microbial habitats, broadly speaking. And then I'll have some time to answer questions on the exam, go over any more slide sets. All right, so that we'll have time for that. So exam is tomorrow. So, okay, we left off here looking at depths of the ocean, primarily looking at deeper depths, how you get this transition where the archaea start to get a little bit of an edge there in terms of better adaptations. This information is just looking at, in general, when you think about deep depths of the ocean, that's fairly extreme conditions that microbes have to deal with. These are some other extreme conditions that I'd like you to think about. Microbes are amazing in their ability to adapt and not just like survive, but actually like do well enough where they can persist, reproduce themselves, and find a home for long term. So some of these ideas, if you look, I would like you to be familiar with some of the category terms that are given. So hyperthermophile, hyper means above, thermophile, think about heat. So we're thinking about the idea that they live at very hot temperatures. Phile means to love. If you're an audiophile, you love music, I think, is how we talk about it. So hyperthermophile, they love high heat. Phile, when they put that part of the word in there, that's emphasizing the idea that they've actually adapted and can reproduce, persist. They're not just able to survive a short stint at that higher temperature. And if you look at that, look at the temperature, defining growth condition, 121 degrees Celsius. Does that ring a bell? What have you learned about in lab that operates at 121 degrees Celsius? It's a piece of equipment we use all the time in microbiology. The autoclave. Yeah, so the autoclave, theoretically, Pyrodictyum could survive. If you popped it in the autoclave, ran a cycle, it would go, yeah, I like this. So kind of amazing. We don't need to really worry about Pyrodictyum contaminating your experiments because its favorite spot is a hydrothermal vent, and we haven't gone down to a hydrothermal vent to collect any of this. But you can see some of these habitats, you know, the hydrothermal vent, submarine vent, hot springs, Lake Vostok beneath the Antarctic ice sheet. You know, this idea of being so cold. Psychrophile is another term I'd like you to know. Psychrophile is cold. You love cold conditions. That's what you're adapted to. Negative 40 degrees Celsius. Definitely cooler than it is today. Acid mine drainage. Iron Mountain, California. Not Iron Mountain, Michigan. So acidophile. You can handle really acidic conditions. pH 0.5. That's even more acidic than your stomach. Halophile, high salt, typically creates a halophilic microbes where they have adapted to that. And so really high concentrations, 5.2 molar salt concentrations in some of these saline lakes. And if you've flown over evaporation ponds out in California, they're usually very beautiful colors, very rich, like orangey brown, and those are microbes hanging out and fully colonizing the really salty evaporation ponds. High alkaline, so if you're alkalophile, you can handle high pHs. So you can see that microbes are incredible with their abilities to carve out niches in areas that would kill us. Here's another one, radiation. Here, they actually hang out in nuclear reactor water in the core. Deinococcus radiodurans was the first one to be discovered when they sampled the water. High gamma, UV, and X-ray radiation. Nothing that we could survive. We would not survive that. Barophile is what we have going on in the deep ocean. So when we think about archaea being able to adapt to deep ocean, if you've taken scuba, you've learned a little bit about pressure. The deeper you go, the higher the pressure on your body. And that also causes certain issues with breathing, of course, because your lungs are getting pressurized too. So have to know how to deal with that, how to come up slowly so you don't get the bends, these types of ideas. So barophilic or barophiles, they're capable of handling that deep pressure. Piezophile is again referring to pressure ideas. Oligotroph means that you can handle low nutrients. So there's many microbes like in Lake Superior. Lake Superior is fairly oligotrophic and especially in the open areas. And so oligotrophs can handle that. Low concentrations. So they're just very good at being able to access nutrients even in low concentration and still take those in and survive. If you're xerophilic, that means that you are in an area where they talk about water activity, which is this idea of available water. So just because there's water around you, it depends on how much solutes are in that water as well. And that influences how readily you can take up water into your cells if you're a microbe. And so this relates to water potential concepts for those of you who've had plant phys, looking at those ideas that if they're xerophilic, they're able to still survive and get access to enough water. They create conditions that makes sure that they can take up water and not have water sucked out of their cells and dehydrate them. Endolith, that's an actual term. Endolith, endo within, lith, rock. So you're actually living in a rock. So if you've ever cracked open some rocks with a hammer and seen some interesting pigments, those might be microbes hanging out. Sometimes they're actually lichens that hang out inside of rocks as well, which is a symbiotic association. So lots of interesting habitats that are extreme from our perspective. And let's go back to the deep pressure. And if you go deep into the ocean, you have some special situations. You have cold temperatures, consistently cold, consistently high pressure, and fairly low nutrient concentration. So if you are a microbe, you have to have adapted to deal with all of that in order to persist. For those of you who might have taken scuba, you know that every 1 atmosphere of pressure, it's every 10 meters down, it's another atmosphere of pressure. And so if you go 1,000 meters, you know that you're getting some pretty good pressure on the cells. So if you are barotolerant, sometimes they'll talk about barotolerant. That means that these microbes are able to go to about 3,000 meters, but when they push them into higher pressures, they don't survive it. Barophilic means they've adapted to really high pressure. So they're deep, deep down. And then the extreme barophiles. So just be aware that they do have like gradations of different groups of microbes in the different levels of the ocean. But yeah, 10,000 meters down, microbes, there's some extreme barophiles. They're fine. We'll talk more about the deep ocean floor a little bit later as well, or when we look at how some microbes are hanging out with some of the other interesting organisms down there. Sometimes they'll graph it out like this, where you see pressure on the x-axis and then the growth rate. And where it peaks with growth rate, that means that's where they're best adapted. So you can see they're trying to show the difference between barotolerant barophile and extreme barophile. And you can see if you push them a little beyond their optimum pressure, they tend to quickly crash in terms of their growth rate. They tend to die. So, of course, that has begged the question, what are they doing to adapt to such extreme pressure? We can't do that ourselves. So when they look at these bacteria, it's challenging to study them because sometimes when you bring them to the surface, they just kind of disintegrate and die. And so they're hard to deal with, but they've managed to get some sampling and handle them in the right way so that they stay intact. And they've looked at their cell membrane and suspecting that they have altered their cell membrane in a way that helps them deal with that high pressure. And sure enough, what they've done is they have put more unsaturated fatty acids in their membrane. So going back to fatty acids, that's part of the phospholipid membrane. Here in the middle in figure B, you're seeing the phospholipid bilayer membrane, and those yellow kinky things are the fatty acids. So if you have unsaturated, like the figure A is showing, it looks like saturated fatty acids. Do you remember from chemistry, if it's unsaturated, what's going on with those fatty acids? If we say, oh, it's unsaturated. Do you remember what that means chemically? Well, yes, in a way. So this is saturated fatty acid. Yeah, unsaturated fatty acids, the notching or bending has to do with the idea that you have double bonds. And when you have a double bond, it creates kinks or bends in the fatty acid tails so that they don't lay next to each other really well. They tend to have a lot more room between each other because of all the bending. And that makes a huge difference, believe it or not. It makes a huge difference. Now, if you put fatty acids, if there's a lot of fatty acids in the membrane that have double bonds, they're unsaturated, and then you have all that extra space between them where they don't lay nicely next to each other, and then you put that under pressure, you can imagine where the membrane can still be flexible, movable, semi-fluid. If it has double bonds, if it has single bonds only, and you put that under pressure, the cell membrane kind of gels up and becomes inflexible and is too rigid for proper functioning. Remember, the cell membrane for a microbe is incredibly important, right? Because they don't have internal organelles. So they have to put all that stuff in their cell membrane. So they need a very nice semi-fluid, flexible membrane to allow for all their reactions, taking things in, getting rid of things out of their cell. So that is one big strategy they've noticed consistently with all the microbes that live at higher pressures. And so that helps them with their membrane transport. They've looked at that. They've noticed some differences to the proteins structure, the three-dimensional structure of proteins that are embedded in the membrane for membrane transport. I'm not going to go into details on that, but they've even looked at protein synthesis patterns and they see some differences there. Proteins in general, the folding of the proteins, the three-dimensional structures are a little different. So they are noticing some biochemical and physical things that the microbes are doing to survive. One of the trade-offs they've noted is that consistently, not even just with deep ocean, pretty much any extreme habitat, the growth rate slows down compared to microbes that are more adapted to this condition of, room temperature and no pressure other than atmospheric pressure. So those types of ideas. So slower growth rate. That holds true for plants too. If you ever wondered why, if you've purchased succulent plants and you're like, man, these things grow slow. Well, yeah, because that's one of their trade-offs. They can survive extreme heat and drought, but one of the trade-offs is it makes them grow slowly. So. So here we're looking at what's going on with adaptations under different extreme conditions. So cold. Again, if you're in the deep ocean, not only is there pressure, but there's cold. And so here they're pointing out some of the things going on with some of the adaptations they've been able to pick up on. If you live in a really hot area, it's kind of the opposite of what we just described in terms of the cell membrane structure. They will actually have saturated fatty acids in their membrane because at high heat, you run the risk that the cell membrane is going to kind of dissolve, so to speak. And so you want a way for it to be a little bit more gelled under heat. And so get your saturated fatty acids in there, and that does help with that. And so here they're just pointing out some key things that they've been studying in terms of how are they adapting to such extremes. With radiation, they actually have really interesting ways to repair any damage to their DNA. Because under extreme radiation, that messes with your DNA profoundly. And they noticed that the ways in which they repair their DNA, it's very rapid and just really ramped up. And so they're very interested in studying that. Okay. Temperature too, we tend to see this idea where when you plot it out and you test different microbes, you have temperature on the x-axis and growth rate on the y, and you can see this skewed bell curve. And that being skewed like that is accurate. What we tend to see is that you have an optimum where you have the peak of the curve. And you can see leading up to that, you can see how gradual this is. So that's saying that at lower temps, they're eking along, they're doing okay, they're able to reproduce. So you see it going up, up, up to the optimum where they're really cooking along at their maximal growth rate. But then you see an interesting dynamic. Instead of a slow gradual decline, if you keep pushing the temperature up, 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 you see this crash. This is pretty standard with microbes that they test. They've tested thousands of microbes like this. And you tend to see this crashing once you push it past the optimum. And that's primarily because you start to really mess with the cell membrane and protein structure. And they can't repair that fast enough. And it ends up pretty severe situation for them. In general, this is a general rule, and certainly there's notable exceptions, but a typical temperature range for a microbe in terms of they're able to persist and be okay, about 30 degrees from their optimum, up to their optimum, but then you push it. Just remember that you push it past the optimum and they tend to tank pretty quickly. So you can plot this out depending on what type of temperature they've adapted to. And you don't have to memorize temperature ranges, but I would like you to know the categories. Like you have the psychrophile, mesophile, we're mesophiles, that's where we fall. Thermophile, hypothermophile. So this gives you some idea of temperature ranges. By the way, there are some psychrophiles, unfortunately, that can, even though their optimum is like 4 degrees, which is your refrigerator temperature, there's some psychrophiles that can be pathogenic to humans. So just be aware of that. So even though we refrigerate food, that's not a guarantee that the food will be safe to eat. There's even microbes that hang out in ice cream that can make you really sick. So that's important, especially if you are a female and pregnant. I was told when I was pregnant with my first child that I couldn't eat ice cream. The doctor told me, try telling a pregnant woman you can't eat ice cream. Doesn't go over well. But there is a microbe, a pathogen that survives just fine in the freezer, but then you eat it and it can cause serious problems for the developing fetus. So There are some exceptions like that where most pathogens are mesophiles like us. They like our body temp, they've adapted to that. But then there's some that can hang out and lurk around in food. They find, of course, I'm blanking on the name of the pathogen. It's a big, long name. Yeah, it caused me a lot of consternation. I couldn't eat ice cream. What was the other thing? Oh, cheese. It shows up in cheese. So it's caused outbreaks over in France, a fair bit, where they have wine and cheese parties and the cheese is contaminated. It'll come to me. I'm trying to access the right neurons there. But anyways, so thermophiles, we generally don't have to worry about, like, those that can survive autoclave temperature, for example. Normally they don't hang around and contaminate our experiments anyways. Bacillus styrothermophilus is one that I've used quite a bit because that is kind of a test for your autoclave. If you pop it in the autoclave, run your autoclave cycle, then you pull it out and plate it out and see if it grows. If it grows, your autoclave failed. So that's another way to test faulty autoclaves. So extremophiles, which we are not, but you can see there's definitely psychrophiles out there that can handle very cold temps and consistently are found when they sample. They take, long core samples of the icebergs, so to speak, in the polar regions. And then, so psychrotolerant means that tolerant is saying, okay, yes, you can push them to a colder temperature, but they actually prefer warmer temps. So there's a difference between phile and tolerant. And you need to pick up on that when you're looking at how they categorize the different microbes. So thermophiles, hypothermophiles, they're there. Think about extreme conditions, volcanoes, hydrothermal vents deep in the ocean, hot springs. Unfortunately, I think some people were getting into trouble with hot springs out west this summer. I heard at least one person fell in and didn't make it. So something like that can kill humans quite quickly, and for microbes, they live happily under those conditions. Can you find extremophiles near you? Yes, if you compost your food. Compost piles, if you've ever turned your compost pile and seen all that heat and steam, you can bet that there's thermophiles hanging out there and are happily breaking down the food you're providing to them. Even just basic soil in sunlight can get quite hot. pH. Again, looking at some ranges here. Acidophiles, if you think especially about soil systems, fungi are very happy in acid conditions. They also are pretty good at surviving low water availability. So if you've ever noticed like your bread, isn't it the worst when you bite into a piece of bread and you taste fungi? I don't know if you can taste, I'm sure you can taste that. It's pretty nasty. It's usually a penicillium that's infecting or contaminating your bread. And bread's pretty low water availability, and yet they're able to handle that. Very often I'll see fungi on the surface of my jam jars, even though they're in the fridge. I'll take the lid off and there's a velvet covering of fungi, because even though there's tons of sugar in that jam, and so there's low water availability, the fungi are fine with that. So not only can they handle typically acidic conditions, but they're good at low water activity. Saltiness. Again, ranges of if you're tolerant of it versus if you've adapted, so you're philic. This one I've worked with quite a bit when I used to teach BI 100, our intro class to non-majors, because I wanted them to visualize DNA and get a grasp of what DNA is. And it's really easy. You just grow halobacterium up in really salty broth, and then you pop them into pure water. And that is way too much of a shift for them, and they just break open, and then you can extract the DNA without doing any serious nasty chemicals, which is good for non-majors. You don't want to give them nasty chemicals and cut them loose in lab. So we worked with it that way. Okay, so these terms, some of the terms you just need to think about a little bit more. Some of them are pretty, if you look at them, it's like, oh yeah, but please get familiar with those terms. Okay, that's the broad training of habitats and some of the ideas on how to describe these microbes in different conditions, especially extreme conditions. I'm going to stop there. We're not going to get into the next slide set because I wanted to leave some time for further questions. Perfect timing. My timer went off. Okay, so let me I think maybe we'll go to where we left off from last time. So that was, I think we were looking at physiology at that point. I think we had brought up this slide set. We're looking, and let me reshare. Okay. Well, certainly key concepts, but I didn't throw anything in there that you have to label because I don't really expect that level of detail. Like I wouldn't want you to label any of this because I'm going for the big picture ideas. But there's a lot of ideas in here. So as you've been studying this, do you have questions on things that need clarification? You're definitely going to get asked questions on this. There's a chunk of the test that has questions on this. So, and the reason for that is I really want you to be comfortable with these big picture ideas because that really helps you to better understand microbes in terms of their ecology, why they show up in certain areas, why they're successful, why they're unsuccessful. So what types of questions do you have? Um, I wouldn't say that necessarily, but let's put it this way. If a microbe has access to hydrogen and it has the tools to use it, they will. Because, I talk a little bit more about this in Gen Micro, how we talk about how easily some chemicals can give up electrons and how other chemicals have a really hard time doing that, so to speak. Hydrogen is way up there. It's super happy to give off electrons to something nearby that wants it. And so if there's hydrogen gas in the environment for the microbe, a lot of microbes have the ability physiologically to work with hydrogen gas and strip electrons off of it. These examples in the table are just some different strategies. So hydrogen gas is one of the top contenders for it. Just is really good at giving off electrons, and the microbe benefits greatly from that. They get a lot of free energy in that process. And then oxygen is kind of on the other end of the scale, where oxygen is just really good at accepting electrons. And that's true for any organism, that's why we use oxygen in our mitochondria. Remember to study these terms too. So slide 32 is this either or idea, how they're paired up in terms of their carbon, energy, and electron ideas, how they're getting access to those. And then you can combine those, so the next two slides are combining those 6 terms into different groups of microbes. Any other questions on this slide set? Okay, well, I'll leave it up in case we do. Methods of sampling is, I mean, there's 3 slide sets on this. Obviously, I'm going to be asking about it. So methods of sampling. So for studying this, yes, you have to be familiar with different like techniques, but I won't ask you details. Like, for example, I won't, like slide 25, I won't say, tell me how to do phospholipid fatty acid analysis. However, I might say, why would you want to do that? And so the next slide is getting at that, like, what's the purpose? What good could it do me in terms of, I have a certain hypothesis or question I'm asking, could that help me answer it? Not tell me the process of it. All right, so when you're looking at these slide sets on methods of sampling, yes, understanding there's different tools and procedures and experimental protocols you can use, but don't get lost in the details of that. It's more like, okay, how would I use this? Why would I use this? What information could it give me that could be useful? So when you're studying these 3 slide sets, it's more approach it like you are a scientist wanting to do some kind of experiment. You have some kind of hypothesis. How would you work through that? Using the information provided in these 3 slide sets. What do you need to think about? What kinds of tools should you use? What kind of experimental protocols should you use? So think about the process of microbial ecology experiments when you're working through this. Questions on this slide set?
The sentence like practicality based, like which test will work best for whatever application we're looking at.
Mhm.
Okay.
Yep, and then like being able to explain why you think that would be the best tool. So it depends on, some experiments, there could be more than one tool that would be totally adequate, but being able to kind of defend your position, like, well, I would choose this because blah, blah, blah. So in other words, this is going to be really hard to do with multiple choice. This is going to be more like an essay because you can't really design good multiple choice to get at what I'm trying to get you to do. So there might be a few multiple choice questions just at some of the basic ideas, but once you get into more of, well, how would you use this for an actual experimental design or process? The second slide set was getting at some other techniques. Again, I'm not going to say, tell me how you clone DNA. That's not the point. Will learn that in other classes, and if you get a job where you have to do that, you certainly will learn, but
It's more like, what could that do for you? How could that be used to help you answer a hypothesis or support or refute a hypothesis? Any questions on any of this content? Yes.
About the stains, most likely to be graded as some questions about.
Yeah, it would be, so like, I won't ask you. Um. Let me pull up that image here. Like slide 22 is showing results from a live dead. The question was how many cells in a sample are living versus dead. And you can do that with acronym orange, but I won't like say, what stain would you use to do live versus dead? But more like, well, if I wanted to figure out live versus dead, there is a stain for that, but you don't have to know exactly which stain, you know? So it's more the thought, the concept of it than the details of it. That answer your question?
Okay, so rather than like specifically pointing out the direct stain type, all we need to say is, hey, it's stain, we know the stain.
Yeah. Yeah. And there's more than just acridine orange out there these days. That's kind of the long, that's been around forever, but there's other stains out there too. So, you know, you would get that training if you ever get in deeper into this, you would know, but at this level, it's more like you understand the concept that, oh, there's stains out there for that. You can do that. Part 3. Where we were looking at species diversity, richness, sampling protocols, pros and cons of that. And then the idea of Koch's postulates in microbial ecology. Questions on any of that? No, I will not ask you to do anything that requires a calculator. No math.
Would it be a lot from the lab? Into this exam?
Not specifically, because you will have a lab exam on that. But certainly concepts you've learned in lab are going to help you with understanding how to approach some of the thoughts on these questions. Remember this last one on the definitions that's posted, that's not anything I will specifically ask you on about. It's just if you wanted that to further help you learn some of the material. And then the populations slide, habitats and populations, we just finished that up, but remember, we're only going through to slide 24. So 1 through 24 will be on the exam. The habitat stuff will not be on the exam.
I think I did point out when we were going over this to know the growth cycle in batch culture. That's like such a standard microbiology thing that you really should be familiar with that idea.
Can you repeat that? What was that one?
The slide 11 with the batch culture, 11 and 12. So being familiar with this concept when you grow microbes in the lab where you don't provide any, additional nutrients or take away their waste. You have that very standard, typical, like pretty much any microbe will do that kind of growth curve under those conditions. And so they've studied that really heavily. It's used all the time. When I grow microbes in the lab, I'm almost always doing batch culture experiments for that. So it's just such a workhorse tool in the micro lab. Which reminds me, so when thinking about the exam tomorrow, I have a pretty solid draft of it. I think there's 30 multiple choice questions. And then 4 essay, 1 is longer and the others are shorter. And it adds all up to 100 points. I think the multiple choice is like 65 points and the essay's 35, I think. Yes.
How many essay questions are getting?
So in class during the exam, there's 4, 1 a little bit longer, the other 3, like, not so bad. You can answer that pretty quickly. The take home right now, I think is 3 questions, 1 big one, 2 smaller ones. I've set it up right now where the take home will start at 11 tomorrow and work through to 9:30 tomorrow, Friday morning.
How long do you have on that?
An hour and 10 minutes. So it's, certainly it's open book, open note. It needs to be in your own words, no AI. And I'll be assessing answers to determine that.
Is this gonna be, is that included in the 120 points?
No,
This is a separate.
I think it's 30 points to take home. It's 30 points, I think.
130.
Yep. Which is pretty typical when I give exams in this class, it's over 100 because we only do 3 exams in this class. I saw another hand. Did you have a question? No, I thought I saw. Other questions? So, yes.
So, we're not doing math equations, we still want us to be able to say like this equation goes with this and this.
No, like memorizing an equation, no. Really the couple equations we had, what was it, we looked at species diversity, Shannon-Weiner equation, and then the other one, carrying capacity idea, looking at that, but no, you don't have to memorize those and write them out. That's not what I'm asking for. So the exam, I'm going to try to get here right at 10 to the hour. I know there's a class before us, but I'm going to try to shuffle them out of here quickly. So if you come here at 10:02 and you get in, I can open up the exam a little early and have you get going on it. And then I'm going to try to stay here as close to 11 as I can, but I know there's a class coming in after us. Then at 11, the window opens for the take home. And it will go through till tomorrow, Friday morning at 9:30. You have lab at 10. Yes,
Clarification, in class portions are locked down
Yes.
outside of class or not.
correct.
No, no, first you can bring.
No, you can have one blank sheet of paper. Sometimes doodling or drawing can help trigger the right part of your brain to help you remember. I'll certainly, the huge reason I sacrifice a lecture period for an exam is so that I can be here with all of you all at once and I can answer questions. So please ask me questions. None of this, I don't do trick questions. None of this is supposed to be tricky, but my brain is my brain. You all have different brains. If I've written it in such a way, you're like, what is she asking? Just ask me for clarification, or if you're unsure, sometimes it might help, like type something up if you're doing an essay. Then call me over and say, is this what you're looking for? And if something's missing or you're on the wrong track, I try to steer you without giving it away. So I'm trying to help you get the right part of your brain going. And because my approach is, you know, this stuff, if you studied, well enough, you know this stuff, but I know what anxiety can do. So if I can help, get that part of your brain triggered, I try to do that. So that's why I like to have us all together. And then the take home, essentially it's open note for that. So it's more like a homework assignment, but it is timed. All right, so I will be looking at my email. I do have office hours today, 2 to 3 still open. My 1 o'clock is full. If that doesn't work, if you want to send me an email, I'll be monitoring my email extra carefully up to the exam tomorrow, okay? Including into the evening.